and reset the position of the bump if it's off. So there is both aspects, you know, this is a recurring theme of our course, this idea there are the interplay between allodetic and idiodetic cues, right? So let's see how exactly this would work. Oh no, okay. Before we get there, let me revisit a slide that we did last week, in which we showed that if you record two head direction cells from the brain, and you know, one is pointing north, okay, and the other is pointing east, right? In that particular room, remember, it's not a real compass. There's no geomagnetic sense. There's no north. The north is arbitrary. Once you get into an environment, you know, that head direction cell might point toward the magnetic north, the geodesic north, the Earth's north, but maybe in another room it's pointing to the south. You know, it's like it resets. It's an arbitrary north. But if one is pointing north, the other is pointing east, like at least that makes sense from, I don't know if you in your camera flips things, but anyways, like that. Then you go to another room. Now this is pointing like this. Well, this is gonna, the angle between the two is preserved. This is what this slide showed, that, you know, you can rotate the cue card, and okay, now you see that both head direction cells, cell 1, my pointer is really slow, cell 1 and cell 2, you see the firing rate of the two curves, right? Now, when you rotate 180 degrees, they both move by the same. Well, I say, well, that's because the cue card, if they both independently they're independently controlled by the cue card, of course, they're both gonna move 180 degrees. It doesn't mean anything about their relationship. However, there are other experiments that show no, I mean, they're always tied to each other. Whatever angle, difference of angle they express, it's conserved, it's preserved everywhere. This is after the cue was removed, and then, you know, they shifted. Notice how the axes here are different. You know, 60 degrees is here, but 60 degrees was here. So it's not the best illustration, but, you know, I don't like the way the axes are, you know, put here. But basically what they did is that they realigned the plots to show that the difference between this and this is the same. So what is this telling us? It's telling us that somehow, whatever network produces this signal, head direction cell signal, and there are network constraints, they're connected. And one way to explain this is exactly the model that we just saw before, right? This model here, you know, you have this plot, this rate map, this polar rate map for one of these cells. But if now, say you record from another cell that is here, well, you're gonna find the curve that is offset by an angle, which is the angle that separates those two cells on the ring, right? And that's gonna be the same everywhere. Wherever you put this system to work, in whatever room, whatever you do, if, you know, the north changes, then all the other cells change their angle accordingly. So that's basically why this model here can explain this property that we saw last week. Let's go one step farther and say, okay, how does it work really? I mean, what makes the bump of activity turn left and right, or rather clockwise or counterclockwise, in a way that reflects the animal movements? What makes that bump? How does that bump of activity know where to move on the ring? Okay, I will say if there are landmarks and you're looking at the landmarks, I make the example, I close my eyes, turn around, maybe I'm off, open my eye, I see the landmarks. Maybe there are particular representation of sensory representations of landmarks that come from, say, neocortex or cortex or sensory cortex, and makes connections on that ring so that it can drive and reset that bump of activity based on landmarks. Okay, but how does we said that that bump is also capable of moving, even if making a lot of errors, moving by itself based on the animal movements, based on idiotetic cues. So for example, your vestibular cues, right? So how does that work? Well, this is what you would do in the theory, right? You would have to have another, at least two rings of neurons, in which basically these neurons on this second ring, let's take this for example, are not only sensitive to where the direction of the animal is, the head of the animal is, but also to angular velocity. Angular velocity. So like your angular velocity, right? You know, you can move around. How fast are you how fast are you spinning and in which direction, right? So if these neurons are sensitive to both the position and the angular speed in one direction, and they are connected in this skewed way, they can move the bump of activity because they're also going to make a bump, this inner circle. But if the animal is moving and now it does have a velocity, an angular velocity in that direction, these cells are active so that you get the activity from this to this and then back to the next neuron. So they are connected in a skewed way so that they can make the bump move in the right direction at the right speed. So they have to be these neurons that are sensitive to both. They're not just pure head direction cells. They're head direction cells that fire when the animal is in a given direction, but also the animal has to move. So without motion on the right, you'll see basically there is just these cells that are the head direction cells that make these other cells fire, or rather they don't make them fire because it's not enough to get these inputs because the animal stands still and so it doesn't have angular velocity. And without the additional excitation, the additional drive that comes from angular velocity, you cannot move. These cells don't become active, and so then they cannot project back the excitations in a skewed way so that to move that bump. So what this model predicts is that there should be cells, I already said that, not just pure head direction cells, but also head direction cells that have these additional sensitivity to angular velocity. And those indeed exist. They are found in one particular nucleus that is connected to the lateral mammillary nuclei that we saw last time. And so there are these cells. So, you know, that's kind of consistent with the model. So that seems to be potentially how these head direction cell signal is created by these called ring attractor. So attractor, I wish we could go more in depth about that. It's called attractor. It comes from dynamic system physics. An attractor in a system, it's a point in the state space of that system that attracts all the trajectory of that system. And so what happens there is that basically it has the fact that there is that bump of attractor, bump of activity and localized activity, reverberatory in one given point is basically what, if you look at it from the standpoint of dynamical system, you would say that that is an attractor. So it would be nice to go in that there, but you know, limits of time and everything, I'm not going to expand too much about that, but I wanted to give you at least a sense. Why are they called attractor? Because basically is the idea is that that activity doesn't move unless there is a reason to move that with these excitations of other inputs. So it's an attractor, it's a stable point in which the activity remains localized there. So indeed, when there is no motion, the activity has to be localized, has to remain there, cannot move around. So that means like there is a state in the system that is an attractor of all the possible evolutions of the system. They remain in that point. I think most of the things we said here I mentioned already. So again, this was a slide from last time. So oh, I forgot to put this. So this would be the dorsal tegmental nucleus. I'm going to add it here. But I mean, the names, you'll find them in the readings that I gave you from the book that were assigned. So I hadn't, but I realized I hadn't put the full names last week. So again, what's happening, it seems like, okay, this is where you find the head direction cells that are also sensitive to velocity, to angular velocity. Lateral mammillary nuclei, you find also head direction cells. Then you move up to the thalamic. So this was hypothalamus, to the thalamic areas. You still have head direction cells. And then to post subiculum, also called dorsal presubiculum, where you also find head direction cells. And I think I said last times that if you, it seems the signal is first generated here and moves up, goes from subcortical to cortical. And because of like delays in how head direction cell represent the direction of the animal. So these tend to anticipate. So it seems like it's where the signal is coming from, where this first, the head direction signal is generated first. And it's probably generated through that, something like a ring attractor integrating the original cues. Sorry, integrating the self-motion, the idiodetic cues, right? And indeed, if you lesion here, then you don't get head direction cells here. If you lesion here, you don't get head direction cells here. But if you lesion here, what happens is that you still find head direction cells here, but they are not capable of using landmarks anymore. And so again, you know, because we rushed last week, you know, I'm kind of bringing back those slides so that we can put them in context of this mechanism that we've been studying. So this was an experiment in which they did standard one was a cylinder with a cue card. You know, we saw so many of these examples. Then they do a second session where the cue card was moved 90 degrees in counterclockwise. And then they did again the same standard two, which was the same position as the first one, right? And so here we have the curves of the tuning of the head direction cell. So you see the black and the blue are almost perfectly matching because of course the cue card was in the same position. So this was a cell that would fire at about, I don't know, 200 degrees maybe, okay? When they turn 90 degrees counterclockwise, of course, the head direction tuning of the cell had also moved 90 degrees because the head direction cell system was reoriented by the cue card moving 90 degrees, right? So very nice. You know, we're seeing basically a system that is responsive to landmarks. So landmarks set the orientation of the head direction cell system. But if you do the same experiment with lesions of the post subiculum or dorsal presubiculum, so they're called both ways because, you know, anatomists don't always agree on how to name things. And so some researchers follow one terminology and other researchers follow another terminology. So you'll see here that there's still curves that are tuned to angles. They're not as clean and nice as the one in the previous plot, but they're still, you know, they're still like kind of a bell-shaped kind of tuning. So these are still, this is still a head direction cell, but you see it's all over the place. You know, you do the experiment, and for every time you put the animal back, there is the head direction cell is still working as a head direction cell, but there's no way to predict what direction is going to fire because the system is not capable of using landmark information. So basically, again, to recapitulate, you know, we have this head direction signal is first generated in subcortical areas, and then, you know, moves via idiodetic cues. Then it reaches these cortical areas like the post-subiculum, where somehow the signal, when I say the signal, the orientation, the head direction represented by these head direction cells is stabilized relative to external landmarks. So that's the job of the cortical areas. Somehow the cortical areas takes this signal that is only produced by path integration from the subcortical areas. Then it gets there, somehow the sensory information comes through for a lot of beautiful reasons that, you know, we're not gonna go through. Like, again, you know, as I mentioned before, like, you know, this is a class that to appreciate it the most, if you already taken a neuroscience class, so you know a little bit about what the rest of the brain does, but, you know, just to say here is that a lot of the sensory processing, you know, can happen definitely in the cortical areas. So perhaps it's not really a mystery. It's not really a big surprise that it's the cortical areas that allow this system to get anchored to the external world, the sensory world. But then somehow these cortical areas provides inputs back into the subcortical areas where, you know, the signal is generated, so that all the system works as a loop and it keeps everything in tandem, you know, like because if you're representing your position on that ring, right, and you only move with the idiodetic cues as a form of path integration, well, we know that eventually we're going to accumulate error, right? So now, but, you know, here comes the post-subiculum, the cortical area that allows landmarks to reset that signal, right? If that correction is not fed back into the subcortical areas, well, now the system is just going to be disconnected, right? There's no way that path integration and landmark information can work in tandem, as we saw, for example, for position system, okay? So I think this is a really beautiful system because it really tells us something about what cortical structure and what subcortical structure, you know, it's a case study. It's a case study where you just see this head direction cell system that almost certainly have something to do with our inner sense of orientation. And they have these very interesting computations that bring together idiodetic cues and allodetic cues. But now we know that there is this kind of division of labor between subcortical area and cortical areas. And, you know, like to study the brain, it's not often that we can really point to a difference of function, a clear distinction of function between subcortical and cortical areas. So obviously we mammals have, as I said, you know, like vertebrates have cortical areas, mammals have neocortical areas, and humans have a lot of neocortex, right? So people are very interested in what does cortex do? I mean, or what does subcortical areas do that cortex doesn't, and so on and so forth, right? So it's really a fundamental question on neuroscience. But then you have these other species, like other vertebrate species, like, I don't know, birds, some crows, sorry, it's my accent. ravens, like those birds can be really, really intelligent and problem solving, and still they don't have neocortex. They have lots of their brain is nucleated, so more similar to these subcortical area, right? Remember those names are, anterodorsal nucleus, all the subcortical structure that are more ancient evolutionarily, they're nucleated. The newer part of the brain is, has a cortical structure. And so it's like there are these other animals, or you might have heard about octopus being extremely smart. Well, the octopus has an invertebrate brain, so it definitely doesn't have cortex. It has all nucleated brain. So it's just really such a fascinating question if you think about it from the neuroscience standpoint, to understand why there are all these different architectures in the brain, that they're different in different animals, and even in the same animal, they coexist, right? Because in our brain, we have neocortex, cortex, subcortex, nucleated parts, it's like it's all there. And somehow there is a divisional labor, but then in some animals, it seems they can do a lot with just one type of architecture. And it seems like intelligence has emerged multiple times in different forms in the evolutionary tree of life in different architectures. I mean, I don't know, if you're a neuroscientist, this is really, one of those questions that are just so interesting, because it's like, why, what, how does it work, so that's kind of big picture question, but so that's why it's so interesting. A direction solve system is like a system that is cognitive, because it's your sense of direction, and it tells you something about how these different parts of the brain that have a different history, they come to work together for a particular function. So let's just say, what do I have here? Well, this is a transition slide. So the idea is that, okay, now we have understood how path integration works in head direction cell system, how landmarks and everything. What can we say about all the other, the cells that we study, place cells, grid cells, especially grid cells? It's like, okay, this works for direction, right? Oh, by the way, there was a last slide, last lecture. I'm not going to go over it because it's pretty clear. It's part of, we didn't have time to go over it, but, it shows how place cells orientation and head direction cells orientation are connected to each other, and that you need a functional head direction cell system in order to have functional place cell system, but it's not the other way around. It's not true. So it seems like our positional system relies on our head direction cell system. But if you lesion place cells, grid cells, head direction cells are fine. They might lose some properties, but they're mostly fine. But if you lesion head direction cell system, all the grid cells, place cell system is disrupted big way, okay? So, but now we can move on. It's okay, can this ring attractor model be extended to position, not just direction, right? And so that's what we're going to look at. And it's kind of similar. So now, instead of having a ring, you have a sheet of neurons. So, like you have neurons kind of organized as a layer, right? And they're all connected. And it's the same principle. Basically, one cell is connected very strongly in an excitatory way to the nearby cells. And then those cells are connected back to it, right? And then there is global inhibition. So now you can have a bump of activity that instead of being on a ring, it's on, a layer of neurons, a sheet of neurons. Again, it doesn't have to be organized anatomically as such. There might be neurons all around in the network, but they have to be connected as if, okay? So, but we illustrate it this way. So now you have a bump of activity that moves around. And here, like you see basically a bump of activity in red that somehow indicates the position of the animal. As the animal moves, that bump of activity moves over the sheet. Okay? And much like, but what makes the bump move though? Well, remember in the ring attractor, we had this other ring, you know, at least for two directions, for the clockwise and counterclockwise, two different rings of neurons that were sensitive to the head direction cell, to the head direction plus angular velocity. And so they could move the bump based on skewed connections. Well, you can do the same. Now you can make all different companion layers of neurons, which are all sensitive to the position, but they're also sensitive to the direction. And then they have like this skewed connection. So if now, I mean, my pointer is so slow, it's just driving me nuts, but it's something about having all these programs open. So anyway, you have the animal here, this bump of, you know, there's this activity here, but because it's moving in this direction, you see that green arrow up there? So now, because it's moving in that direction, only the cells that are on these layers that is sensitive to that direction are also active. And because of the way they're connected in a skewed way, now they're gonna make that bump move. And you know, here's the illustration. And then if there is no motion, this is the only layer that is active. So there is one layer that is purely positional. And there are all these other layers that are position times, it's conjunctive. It's called a conjunctive encoding, a conjunction, right? And a conjunction is and, means logical end, right? Conjunctively encodes the position and one particular direction. So you need many because you have more directions. So based on the direction of the animal, one of those are active. And if the animal is moving and these neurons are sensitive to the head direction and velocity, then they can move the bump around. So it's just the same principle, okay? And so if you were recording from a cell on this layer, well, you would see a place cell, right? And if you were recording from one of these layers, well, you will see a place cell that is also looks like a head direction cell. So this cell only fires when the animal is in this position, but also conjunctively. So both have to be true conjunction. The animal has to be pointing in one direction, and there also has to be speed in order for all these systems to work, right? So this is what would you predict. And indeed, there are cells that have that, there are place cells and sensitive to also the head direction cells. We'll look at it in a little bit. But, you know, a ring, because the angle is a circular variable, right? On the ring, we didn't have the problem of what happens when you get to an end because there's no end. You always turns around, you know, your direction is circular, but here we have boundaries. So what happens if the rat keeps walking, at some point it should fall off that sheet of neurons, right? You know, you have, like at some point you gotta hit the limit, you know, like you only have so many neurons. So whatever this system works is gonna have that limit. And so what happens here? So how would the system work? Okay, well, one solution is to have kind of the equivalent of a ring, but in 2D, right? So like this sheet of neuron has to wrap around in a complicated way, which is really not that complicated when you think about it. It has to be to wrap around in both directions, like the two main, you know, if you think about movements in space of the animal on a 2D environment, like on the floor of a room, then, you know, you take two orthogonal axes, like X and Y. Along those two axes, this sheet has to wrap around. By wrapping around, again, it's only a matter of connection between cells, right? So this is, again, it's not necessarily a physical sheet, it's just the way that the cells are connected. So you need to basically, when you get to the point, to the end, you need to be connected back on this other end. So basically, it's just like this, instead of being a sheet of neurons, it becomes a donut. So you close it in one direction, you wrap it in one direction, and then you close the whole thing on the other side. So as the animal moves in 2D, those are the two directions, you know, the bump of activity, the localized bump of activity is moving on that donut. It's called a toroid, a toroidal structure, right? So now what happens if you're recording from one of these cells? Remember what we did? We're doing, basically, we are doing the same thing that we did for the head direction cell scene, extended to position instead of angles. So remember, you know, say, okay, if I'm now recording from one of those cells on the ring of the head direction cell, that's gonna look exactly like a head direction cell. What would this cell, you know, if you record one of these cells, look like? Well, as the rat moves around, the bump of activity is gonna move. At some point in a certain position, the bump of activity will pass through that cell, so the cell will be active. So it's gonna look like a place cell. However, because this is wrapping around, if the animal keeps moving, eventually it comes back, the bump of activity comes back to the same spot, right? It's like a ring, but now there's two dimensions of it. So what you would see if you were recording from one of these neurons is a grid, but not an hexagonal grid, a square grid, right? So if you were recording from one of these neurons, as the rat moves around, now because things are wrapped around, like if the rat, say, is moving in this direction, it can go all the way around the donut, then the bump of activity, so the rat keeps moving, right? The rat keeps moving, but now the cell is firing again. So if the rat is moving from here, it moves, the bump of activity can go around, so different cells are going to be active, but then when it comes all the way back here, the cell is active again. So what you're going to see in a rate map is a second field. It's a second firing field, a second place field. And then again and again. In this other direction, you're going to make, you're going to go around the transversal section of the donut, right? So same thing, you know, you go around the transversal section of the donut. Now it means you're moving in this other direction. And so it's like now, you move, and again, the cell fires again, even if now the animal is in a different position. So you're going to have a regular grid, but now this grid is square. And there, you know, when we record grid cells, the grid is hexagonal. It's not a square. It's just, it's not, you know, like there's not such a thing. So this model is wrong and can be rescued. Does it still say something that perhaps, you know, is how they're organized? Well, if you take a donut like this and the connection, you skew them in a way that is, the donut is twisted, like kind of like this, because it's twisted. Now you need to do a stretch of imagination and really think about it. But because it's twisted now, when the animal moves around, and you know, it's not going to be a square grid, it's going to be an hexagonal grid. And so one of the prevailing model for how grid cells, why grid cells exist, why a cell fires in these hexagonal ways, such, you know, what you know, that's what we observe. What is it telling us about the brain? And the possibilities that your representation of position is kept track through path integration, through, like, you know, speed and direction over something that is organized as a donut. And because of that, so that you don't have the problem of falling off the word, because you don't have that problem, but then you have a repeating field, and that's why grid cells are grid cells. And maybe you have, of course, you're gonna have different donuts that are tuned differently to speed so that the grid will be, we're gonna get the grids that are more spaced, the grids that are less spaced. That's all based on how quickly the bump of activity moves on that donut when you're moving. So I'm moving, say, 1 meter, how much does the bump of activity moves on that donut? If it doesn't move very much, it will take a longer time for the bump of activity, or rather, it will take me many more steps. I would have to walk much longer before that bump of activity completes a revolution around the donut and the cell fires again. So the grid cell at that point is going to have a larger scale. If the bump of activity moves faster, rather, as I move, it moves more, then it will take me fewer steps before the bump of activity makes a full revolution around the donut and the cell fires again. So you're gonna have a grid cell with short spacing between the vertices, kind of like this one, for example, right? So again, though, you will need cells that have sensitive to both direction and position in order to move this bump based on the sense of velocity, based on path integration. This one I've just put here is just another way of seeing things. This starts from the grid. And so you say, okay, if we get the grid and you cut the unit base or repeating pattern like this, and you have this distance between the farthest fields here, now you can wrap it around in one direction, then in both directions. And now you see that path from one field to another becomes a path that makes a revolution around the donut in both directions, right? So it's going to go around the donut, but also around the section of the donut. So that is why the torus has to be twisted if you want to map linear movements of the animal over a twisted trajectory like this, right? So it's just a different way to look at this, if it helps you. But if we go back, remember, because we have the ring attractor, we have this property, all right, maybe I shouldn't have done this, where the two head direction cells preserve their angular differences, right? On the donut, you have something similar, so that if you're recording from two cells on the donut, because the bump moves in that way, when you're gonna look at those two grid cells, those grid cells will always have a firing rate map that is offset from each other by the same vector. So the way that the grid pattern is offset from cell to cell is always the same. That is the property that we studied before, right? Remember? And the idea that if you take multiple grid cells, at least of the same scale, the phase might change in different environment, but the difference in phases, how offset they are, it never changes. So they preserve their positional relationship to each other. And that is also a consequence of this model, of the model with the theoretical model based on a toroidal network, attractor network, just as much as the angular consistency of head direction cells was a consequence of the ring attractor. So this is why, this is one class of models for um, um, these. Oh, one thing that I probably should change the order of these slides, but as we said, for the system to work, you need to have those additional cells that have sensitivity to position and direction and speed, right? And on a ring, on the ring attractor for head direction cells, we put 3 rings. For the position system, we saw all these different layers. One is pure position, the other are position and different directions. Then we took that layer and we made a donut of it. Well, you would have to make donuts of the other ones. It starts becoming a little complicated to show, so we haven't showed it. But still, the model predicts that there should be cells in the brain that have the fire as a grid cell, but only when the animal is pointing in a certain direction. So, and guess what? There are these types of cells. So, you know, presubiculum and deep layers of MEC, you can see, okay, let's look at this grid cell, right? It fires with three vertices, but if you look at the polar plot of that, based on that direction, is very pointy, meaning this is at the same time, conjunctively, it's a head direction cell and it's a grid cell too. What it means is that it's not true that the cell fires every time the animal is walking on the spot here. Only those times when the animal is there, but also pointing in a particular direction. So remember, there is this conjunctive representation. Conjunctive means and, so it has to, both things have to be true. The animal has to be in this position and has to be pointing in a certain direction. And so these cells have been reported. These cells here in presubiculum, chances are that those are also grid cells. It's just that people were traditionally recording from a smaller environment. And as you know, if its environment is small, you're not gonna see the grid. You're gonna see only one vertex. So if you were looking at the polar plot for a normal grid cell, you wouldn't find the droplet shape like this. You would find like a circle because the cell fires every time the animal goes back on that vertex, regardless of direction. Remember what we said, place cells fire regardless of direction, unless the animal is on a linear track, on a one-dimensional track, then they're sensitive to direction, right? For reasons that we still don't understand. Okay, so, you know, lots of things that come out together beautifully. There is another major class of models that is a competing hypothesis, a competing theory. And we're gonna see it at in our last lecture when we talk about time, because that's a model that in a way it is turning oscillations in time, similar to the one that we see with theta, into oscillations in space, which are grid cells. And so that is where time is one way that time comes into all this. Well, in a way, it's already coming, right? Because when you're doing path integration, if you think about it, you're turning velocity into position, right? You're estimating the update of position based on velocity. You remember from calculus and physics that that's an integral of, if you do the integral of velocity, the integral in time, well, you get position, right? Because velocity is the derivative in time of position of space. So to get a position from the velocity, you have to do basically what is an integral in time. So that's also like it's path integration, right? So I managed to lose my train of thoughts here because I opened these parentheses and yeah, so there is this other class of models that have to do with time. And so I'll keep them for our last official lectures in 2 weeks, okay? Rihanna, you can unmute yourself and ask your question, since I see.
Yes. So I heard the word integral. Will we have to know how to calculate that, or do we just need to know that the integral of, I think you said velocity or something like that? Do we have to know the math behind it or no?
No, I mean, there's not going to be math. It
Okay.
helps to know, that's to remember from high school physics that the derivative of space is velocity, and derivative of position is velocity. Integral of velocity becomes position. So in a way, that's what's happening here, right?
Okay,
So
thank you.
and um, so there is a competing class of models. There is other models as well. We don't know how all this works, but this is where our thinking is at in science about all these mystifying phenomena. So let's see. Yeah, so this is basically what I just said. All right, last 10 minutes, I'm just gonna give you a little bit of, this is a little bit of a bonus here. So a lot of these things that we're studying, they are a problem for engineers that work with robots. If you make mobile robots that go around, like the newest generation of the Roomba, for example, makes a map of your environment, right? The algorithms, the system that engineers put in these robots have to do with path integration, landmarks information. So they're basically facing the same problems that the brain is facing. That's actually my history. I was in AI, I was just working on those systems in robotics. That's how I kind of got interested in how does the brain do it? And then I switched to neuroscience. I might have said something about like in the intro lecture. Anyways, I don't want to make this about me, but I want you to just like appreciate that there is a certain interdisciplinarity about these issues, because the same issues confronting engineers. And if you've done that, you realize how difficult this problem is to make calculations, and you wonder like, how does the brain do it? So like here it's just like, okay, you have a maze and you have a robot that moves around in the maze. On the left, you have the actual trajectory of the robot. On the right, you have what the trajectory would be computed in the software of the robot if you could only use what is called odometry. You know what an odometer is, right? It's that thing that tells you how far you've traveled on your car. It counts, how many times the tires rotates, the wheel turns, like an estimate of the size of the wheel, or other systems, right? It's like the equivalent of step counting. The odometer of your car is a system that uses an idiotetic cue, basically. It's not, if you think about your car as an organism, your odometer is basically self-motion. It's like path integration. So how do you correct that? Because there is all this error, right? You got to use landmarks, and people use very complex algorithms. This was work that I was doing. You get a mobile robot that goes around and, in an office space, and you want to build something like a map of empty space. The white is empty space, correct? But because you make all those automatic error, you can end up with a map like this. It's hard to connect loops and close loops and know when you're back at the same position where you were before, unless you have a complex system of balancing what your path integrator is telling you and what the landmark information outside are telling you. And you have to use very sophisticated probabilistic algorithms. And so that was like one of the things that got me into, was like, well, somehow the Placetel Gritzel system is able to do that. And that might be a way to kind of use it as a Rosetta Stone to understand how the brain computes, right? So these are, computed through kind of similar to a sonar. Also, it's done with a laser, so it finds the range. And it has a lot of applications indoors. As I said, your Roomba, lots of unmanned autonomous vehicles in space exploration, mine exploration. So like the old mines, sometimes you have to explore them. It's better to send a robot than a person because it's dangerous. And lots of applications for this stuff, right? And so this issue of how to make a map is a big problem in robotics. Here I put a couple of YouTube videos. You can look at them. It shows how a robot can go around, start making a map, and then when he comes back and he closes a loop, recognizes that it's back to where it was before, and that allows a correction of the map because it can correct all that path integration error that had accumulated around that loop. So going back to the same position and recognizing that you're back, if you're off in your mind, in the mind of the robot, now you can correct that. So somehow, everything we're studying is the brain doing these things to some extent, okay? Just wanted to put like 2 or 3 slides of that to just kind of get this interdisciplinary thing and just kind of interdisciplinary appreciation of everything we've been doing and that we keep doing. So that would be it for today. I'm here to field any question. And I mean, if you don't have questions, you can log out and leave. You're free to go. If you have questions, I'll stick around. I hope it was an interesting lecture. The presentations, I'll send the date. I mean, their presentations are, stop sharing here. Give me a second. It's going to be starting from November 9. So in about, what would be like 2-3 weeks from now. So there's time, and that's the first day. And then it's going to be probably so November 9, November 23, and November 30. So by the end of this week, I'll assign the reading. I will give the dates, and I will share a little documents with advice on how to prepare these presentations. No. So Thanksgiving, let's see. So Thanksgiving is, well, we're gonna have the second test the week before Thanksgiving. And then, let's see. We're gonna have I believe we're gonna have the review. So it's going to be, the student presentation will be, yes,